Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my YouTube channel, Science for Everyone. Today I will discuss a very important practical that is related to bioinformatics. Okay, and the practical name is how to perform molecular docking. In the previous video, I have discussed that what really molecular docking is and what are the steps that we will follow and we will perform molecular docking. Okay, so here I will not discuss the steps. Okay, I will just perform the steps. Okay, and just take out a page and a pencil with you. Okay, so then you can write your these steps. Okay, if you haven't watched the molecular docking video, okay, so I recommend that first to watch that video first. Okay, so then you should have an idea that what really molecular docking is. Okay, so then it will help you in your assignments, in your presentations. Okay, and in your exams. Okay, so let's start with the practical. Okay, I will perform these steps very smoothly and in a very slowly. Okay, so and this practical is a bit we can say it is a bit longer practical. Okay, so it will take time. Okay, so don't get bored. Okay, and just follow the steps. Okay, because we first we will do different steps. Okay, we will download the macromolecule, the target molecule, the ligand, and after that we will perform docking. Okay, so it will take time. Okay, so let's start with the practical, and don't worry, it's too easy. Okay, okay, so first the requirement of molecular docking is that we should have a macromolecule, a protein, and then we should take a ligand. Okay, it should be a drug. Okay, and after that we will dock these two molecules, and then we will see that how much efficiently they have bind to each other. Okay, so first of all, what we will do, we will download a macromolecule. Okay, so what is the process? We will go to the Google Chrome. Okay, and we will type protein data bank. Okay, I have typed it earlier, so it is in my recommendation. Okay, so you will type protein data bank and you will enter. So this is the home page of protein data bank. Okay, here you can download different proteins. Okay, so you according to your examiner, or you have a protein is given to you. For example, it is we can say reverse transcriptase. Okay, reverse transcriptase. Okay, so here is the recommendation. Okay, so this is a reverse transcriptase molecule. Okay, a protein. Okay, so you have to click on this. Okay, after this wait a minute. Okay, you first of all you have to click on this 1 mml. Okay, so here is uh, the picture of it. Okay, wait a minute, it is taking time. Okay, so you can also click here, download file. Okay, and pdb format. Okay, protein data bank format. Okay, because when you are performing docking, okay, so it must be in the pdb format. So you will click on the pdb, and here your protein will be downloaded. Okay, so now you can open your protein. It will be present in the downloads. Okay, here is the downloads. Okay, so this is the protein. Okay, so I'm having a chimera software. Okay, you should have chimera with you people. Okay, chimera 1.15, it should be downloaded on your computer. Okay, because on your laptop or your computer. Okay, so it is very necessary for this protein to view it. Okay, you can also use discovery studio, but this chimera is very much simple and easy. Okay, so you will click on your protein. Okay, so chimera is open and your protein is just like this. Okay, just look to the protein. This is reverse transcriptase that you have downloaded. Okay, so I want to tell you guys that what will you do. Okay, so according to the literature, if you have studied the literature and the active site of it. Okay, so many protein have many subunits. Okay, here the reverse transcriptase have only one subunit. It is okay for it. If they are having many subunits, okay, so then you can delete the subunits. Okay, and you will keep the desired subunit. Okay, but here we are having just for this our one subunit. Okay, so we are okay with it. Okay, so what we will do, I want to tell you something. Okay, this protein is in the protein data bank. It is in such a way that the water molecules and the other essential molecules, when it is present inside a cell, it is not bonded when it is present in protein data bank. Simply, अगर मैं आपको बता दूं, so in the cell, our this inside the virus, the reverse transcriptase protein is present in such a way. Okay, there different water molecules and different other essential molecules are binded to it, but in the PDB format it is not binded to it. So what we will do, you will go to the you have to make your protein just like when it is present inside the cell. So what you will do, you will click on the tools. Okay, and you will do structure editing. Okay, and what we will do, you will minimize your structure. Okay, so it will be just like when it is present inside the cell. Okay, so you will click on the minimize. Okay, so I want to tell you that it will take much time. Okay, it will you will minimize, you will click on minimize, and after that you will wait for it. Okay, so and just press on the okay. Okay, okay, again. Okay, okay, so then docking preparation finished, and after that these steps will be performed. 100 steps of steepest descent. Okay, so 10 of out of 100, so it will take time. Okay, so we will then I will just pause the video, and when it is completed then I will run it out. Okay, okay, now our structure is minimized. Okay, it is just like the structure that is present inside the cell. Okay, of the virus. Okay, because it is a reverse transcriptase. Okay, so it is just like the protein that is present inside the virus. Okay, so now what we will do, we will select on the file. Okay, and we will save it save PDB. Okay, we will click on the save PDB. Okay, so you will click on the change drive. Okay, so where you have to save it. Okay, you will just click on, I'm saving it in the D. Okay, so you will click on the D, or if I want to save it in the E. Okay, so okay, D. Okay, okay, now the file name, so then you can call it as a you can just write as a reverse transcriptase or a molecule, your choice. Okay, then I'm giving it reverse. Okay, and dot pdb. Okay, pdb. I don't know, I have told by my professor that you have to type pdb. Okay, it is important. Okay, after that then you have to click on the save. Okay, so your structure is saved, so your molecule, the this molecule is now saved on your device. Okay, so you have to cross it. Okay, if you want to see it, so just click on the my computer and D. Okay, so here is your molecule. It is now just as required by the doc for the docking. Okay, now you have to take the drug. Okay, okay, you have to take the drug. Okay, so you have to click on the drug bank. Okay, in the Google, you can take it from the zinc data bank or anything else, but the drug bank is very much easier. Okay, here you can type your drug name. Okay, so any type of drug structures are present here. Okay, so I'm clicking on the streptomycin, for example, and then click enter. And after that what will happen, that you have to download it. Okay, so what we'll do, we will just click on the download, and you will download it as a pdb. Okay, we have downloaded as a pdb. So here is a what we will do, we will control a, we will select it all, we will copy it. Okay, just follow the steps. Okay, we will copy it, and my pc. Okay, uh, go to the D, create a new text document. Okay, and after that open it, and you have to paste this. Okay, you paste it, and you click on the file and save it. Okay, so now cross it. Okay, what we will do, we will open CHIMERA. Okay, CHIMERA software, it should be downloaded on your computer. Okay, you will click on the file, you will click on open, Okay, now you have to save it in the D. Okay, so you will click on the D. So here your new text document is here. Okay, you will click on it. Okay, and you have downloaded from the PDB format. Okay, so here you will click on the PDB. Okay, this is the PDB. You have, okay, you have to click it. So here is your streptomycin drug. Okay, so now what you will do, as the same you have done for the molecule. Okay, so you will now do it for this also, that you will click on the tools, you will do structure editing, you will minimize the structure. Okay, minimize it. Okay, and then you will click on the okay. Okay, so just wait for it and then click on the close. Okay, so now your structure is as the same as it is inside the cell when a person eat this streptomycin. So how it will act, and these are the, these white were first not present, now it is present. These are the water molecules or the hydrogen bonds when it is present inside the cell. What you will do, you will click on the file. Okay, you will save it as a save pdb. Okay, after that, where have you you want to save it, so I want to save it in the D, and I want to give it a name as a uh, strepto. Okay, so it is now, and you have to pdb, you have to type it. Okay, and after that you have to save it. Okay, so now you your macromolecule, this is the macromolecule. Okay, this is the macromolecule, this dot enzyme, and this is the streptomycin. So now these steps are completed. You have gathered your items for the docking. Okay, now what you will do, you will have to click on the Pyrex. Okay, this is a Pyrex software. Okay, you have to download it. I think it is 400 to 200 MB. Okay, you have to download it on your computer. Okay, so you have to click on the Pyrex. Okay, so this is a software used for molecular docking. Okay, so it will take a little time. Okay. Okay, so this is the Pyrex software. Okay, this is the front page. Here you will perform the docking. Okay, so you will click on the start. Okay, start, and you will click on the auto dock. Okay, one is the Vina visit, and one is the auto dock. So the auto dock is a bit easier. So you have to click on the auto dock. Now you have to select the molecules. Okay, so select molecule. First you have to click on the add macromolecule, that reverse transcriptase. Okay, you have to click it. Okay, and after that where have you saved it? Okay, so you have saved it in the D. Okay, so here is the D, and this is the reverse transcriptase. Okay, so you have to click open. Okay, so after that it will take a minute or two, or even it can be uh, be faster. Okay, so here is your molecule. Okay, you can check it. Okay, so here is your molecule. Now after that you have to click on the add ligand. Okay, and your ligand is in the D disk, and you have to click on the strepto. Okay, this is the ligand. Okay, so now this is our ligand. Okay, and this is our macromolecule. Okay, so our steps are now going to completion. Okay, we have to just okay, this is the strepto, and this is the reverse transcriptase. Okay, so now I want to tell you guys that I have told you that in the literature or from other softwares, you have to find out that where is my active site. Okay, for example, we are choosing our active site. Okay, so you have to click on this plus. Okay, so for example, this proline 30 is our active site. Okay, so you have to right click over it, display label and atoms. Okay, so here you can search for it. Okay, there is a bit can say here a labeling is done. Okay, for example, if we do it for glycine. Okay, display label and atoms. Okay, so here, okay, just look to it. Here it is, the labeling is done. Okay, for example, this is our active site. Okay, if I want to roam it around, so then you can see it easily. This is our active site, for example. Okay, So now what you will do, you have choose your active site, you have choose your macromolecule, your ligand. Okay, after that what you will do, you will click on the auto dock. Now you have to perform the docking. Okay, so what you will do, you will choose your you will click on the press the control, and you have to choose the reverse and the legend. Okay, and you have to click on the forward. Okay, sorry, I have not select the legend. Okay, so this is my legend, and this is my molecule. This wait a minute. Okay, so then you will click on the forward. Okay, so a box will appear. Okay, just look to the box. Okay, this is a box. Okay, a box will appear. You can drag this box and you can place it over the selected area, this white area, because you have, this is your active site, for example. Okay, so you can just put it over it. Okay, you can also minimize and maximize the size of this box. Okay, so here, for example, this is the active site of you. Okay, and after that what you will do, you have select your active site, and you are, then you have place the box over it. Then you have to click on the forward. Okay, when you click on the forward, so then it will take time. Okay, because it is performing sorry, my this virtual skinning tool is not responding. Just wait for it. Okay, okay, so I have clicked on the forward. Okay, so this is now the result. Okay, just wait a minute, I want to show you in a clear way. Okay, so this is the result. This was our molecule. Okay, and here I have performed docking. Okay, my receptor molecule, my ligand is now docked with this here. Okay, so this is now the result. Okay, so don't look to the these result. Okay, this is not for of our concern, just this is for of our concern. Okay, I want to tell you guys that I have changed my the reverse this reverse transcriptase name as MOLE. Okay, it was a bit easier for me. Okay, and I have changed the streptomycin, that strepto, as a DDD. Okay, I was having different files, so that's why. Okay, so this was our molecule, and this is our drug, this streptomycin, and this is our reverse transcriptase, and this is the binding affinity. Okay, minus 6.7, minus 6.3. Okay, so I want to tell you guys that what does this means. Okay, so just simply remember. Okay, if it is below minus 10, okay, now it is minus 6.7. If it is below if it is I think minus greater than minus 10. Okay, or so sorry, lower than minus 10, if it is minus 11 or minus 10.5 or minus 12, so it, then that binding affinity will be much more efficient for us. Okay, the greater the value, the lower will be affinity. For example, it is minus 5 or minus 3 or minus 2, so then it will having a very much lower affinity. Okay, so the more the lesser the value, okay, the more the value is in minus. Okay, the more our binding affinity will be. So here it is minus 6.7, so it is not a good affinity. Okay, a good binding affinity. So our this drug cannot be efficient for against this reverse transcriptase. Okay, and we all know that we are not giving a patient if it is a it is an if a patient is HIV infected, so we are not giving it streptomycin against it. Okay, we all know it. Okay, because it is an antibiotic, and that is an antiviral drug. Okay, so if you perform an experiment this practical over reverse transcriptase, and you take a drug that is stuatidine. Okay, that is an antiviral drug. Okay, and it acts against HIV, so then I think it will show you minus 10 or minus 11. Okay, so perform it, just follow these steps, it's easy. Okay, I want to tell you guys that this Pyrex software is a bit it is lagging. Okay, the best software is the gold software, but it is paid software and it is very much expensive. Okay, and this Pyrex software is a bit it has no cost. Okay, so you can use it, but it can take your time because this software is very much lagging. Okay, so just keep this thing in mind. If it is lagging, so just cross it and just play it again. Okay, run it again, so it will work. Okay, there is if you are not knowing the active site of your enzyme. Okay, so it might then give you false result, or it can easily give you an error. Okay, if you are not getting your best active site. Okay, so then it can give you a error. Okay, so keep this thing in mind. If you it is giving you errors, okay, so don't need to get panic. Okay, because you are not knowing about the active site. There is a software where you can find the active site, or you can also find the active site on the Google. Just write the enzyme name and just ask the question that where does the active site lies. Okay, so then you can easily select it and you can then easily dock your element and your ligand over it. Okay, so this is the active site. Okay, so I'll just dock my streptomycin over it. Okay, so it gives me -6.7 and -6. Okay, all the values are in above -6. Okay, so it is not a good binding affinity. If it is lesser than -10, okay, -11, -12, -15, so then it is a very fantastic drug for your enzyme, for your antigen. Okay, so thank you very much. It was a very it was a bit detailed practical. Okay, if you have any question, you can ask me in the comment box. Okay, I will answer you there. If you are having any problems related to this practical. Okay, so this was all about molecular docking. If you haven't watched my lecture of molecular docking, so first watch that video. Okay, so then it will be easier for you to just perform this practical. Okay, so thank you very much. God bless you.